Alzheimer's carries a stark statistical weight. Current estimates place the lifetime risk at 1 in 10 for males and 1 in 5 for females. When a relative is diagnosed, the immediate question is personal. Most people look at their family tree and wonder if they've inherited a biological countdown. But the assumption that Alzheimer's follows a single genetic path is a misconception. Inheritance actually follows two separate tracks. One is a locked biological switch. The other is a complex game of genetic probability. Understanding your personal risk depends entirely on identifying which of these two tracks you are looking at. The first track is the early-onset form. It accounts for less than 1% of all cases and typically appears between the ages of 30 and 60. This track is driven by mutations in 3 specific genes: APP, PSEN1, and PSEN2. These follow an autosomal dominant pattern. Inheriting a single mutated copy from one parent guarantees the disease will manifest. In clinical terms, these mutations are fully penetrant. These mutations act as faulty cellular scissors. They miscut proteins, causing an overproduction of toxic amyloid beta that rapidly clumps together in the brain. For this rare group of patients, inheritance is destiny. Environmental factors and lifestyle choices have effectively no impact on the outcome. Late-onset Alzheimer's follows a different logic. This track represents 99% of cases, and here genetics act as a susceptibility dial. The most significant factor on this track is the APOE gene, located on chromosome 19. APOE exists in 3 variations. The E3 allele is the neutral baseline. The E2 version is protective, turning the risk dial down. But the E4 version is a risk multiplier. Carrying one copy of E4 increases risk 3 to 4 times. Inheriting 2 copies, one from each parent, spikes the odds to a 12-fold increase. Crucially, APOE is a susceptibility gene, not a determinative one. Because it is semi-penetrant, even someone with 2 copies of E4 is not certain to develop dementia. Other variables turn this dial up or down. For instance, the impact of E4 is significantly higher in women, particularly between the ages of 60 and 70. External modifiers play a role too. The E4 effect is most pronounced in those of East Asian ancestry and is heavily worsened by vascular issues like smoking, diabetes, and hypertension. Late-onset Alzheimer's is a probabilistic equation. APOE sets a baseline, but sex, ancestry, and lifestyle determine the final odds. This uncertainty creates a clinical dilemma. Because there were no ways to prevent the disease, doctors traditionally discouraged testing for the APOE risk gene. Clinical practice is changing because of new anti-amyloid therapies. These treatments carry severe risks, like brain hemorrhages or swelling, that occur much more frequently in patients carrying the E4 allele. APOE doesn't explain every case. To find the missing heritability, scientists are conducting massive genome-wide studies, analyzing the DNA of hundreds of thousands of people. These studies have identified over 80 other risk genes. Rare variants like TREM2 have shifted the research focus toward the brain's immune system and how it clears waste. The genetic map of this disease is proving to be much larger than just amyloid or APOE. It reflects a multi-system biological breakdown. Alzheimer's presents a dual-track reality. It is either a rare, predetermined biological event or a common, modifiable probability. For the vast majority of people, DNA does not dictate an unavoidable future of cognitive decline. Decoding these genes provides a roadmap of the biological mechanisms, like lipid processing and immune regulation, that drive the disease. Deciphering this map isn't just about prediction. It is the foundational blueprint we need to eventually develop a cure.